So I'm very thankful for Louisiana Sea Grant and all of the opportunities that they provide. I'm getting to work with many stakeholders, many people who are involved with Louisiana Sea Grant, and their impact, I think, across Louisiana and the United States as a whole is very big. So what is white spot syndrome virus disease? It's a long word, so I abbreviate it as WSSV. So it is a disease that's been found in wild and cultured populations in multiple species, not just crawfish, shrimp, crabs, lobsters, other copepods, and it's thought that many organisms are infected by this virus, but some might not see the effects that we're seeing in crawfish. So there's um, in these crawfish, the populations can be completely wiped out, which is absolutely terrible for the industry and for these farmers. But we're not seeing this in all organisms that might be infected by the disease. So, like I said earlier, this was first kind of brought to the of the front of the docket in 2007, but there were some pretty hard hits in Louisiana in 2017. So, in talking with farmers, they saw a lot of loss starting 2016, 2017. The industry was increasing, production was increasing, and this was a pretty hard hit for them. So why is this important to study? Well, there is kind of a lack of understanding of the different strains of the virus. We're working to test if how crawfish get the virus, how they're susceptible, and what kind of factors play a role in that. So there's a lot of people on this project, great researchers who are doing awesome work, and I'm just one of the ones playing a part in this. So I'm looking at the environmental factors rather than sequencing the viral genome of this. There's people at the vet school and in renewable natural resources who are working on that side of the project, kind of a micro scale, while I'm more looking at the environmental side. So with that, I'm interested in these abiotic factors such as temperature and precipitation and things that are going on that could lead to outbreaks in these ponds. So I really am trying to understand practical environmental drivers and how maybe this is transmitted pond to pond. Does it have anything to do with change in weather, change in water temperature? And is this something that we can control? Is this something that we can show farmers how to manage for and maybe bring back the effects of this virus? So why do we care about crawfish? Well, of course, many people do here in Louisiana, but there's more reasons just to care rather than eating them. They are delicious. So ecologically, they start as small little guys. They're pretty cute, I think. I might be biased, but when you think about this little organism ecologically, there's a lot to think about. So in the food web of a crawfish pond, they're eating decomposing plant matter and living plant matter, all of these microorganisms and epiphytic organisms, and it's really important. It's a really important cycle. There's vegetation in these ponds. Some farmers grow rice, and so that provides a structure of the food web for these guys. So it really is a whole cycle that sometimes we don't think about when we're eating them with butter. And so what's also cool about these organisms is that they have natural recruitment from mature animals to populate these ponds. So it is a pretty in-depth cycle, more than you might think. So of course, culturally, crawfish boils in Louisiana are a huge social event for people who enjoy eating them, but also for farmers and their families. This is their livelihood. Most of these farmers have been doing this for generations. And so this really is a culture for them and their families. And if you ever get to visit any of these parishes in South Louisiana, the farmers will make sure and tell you how important these crawfish are. So another big one, economics. There are over 1,500 farm operations and over 31 parishes across the state, which is huge. There's so many jobs in production and processing and the supporting industries of distribution. And in 2016 alone, millions of dollars were in this industry. So this is a big deal, which people in Louisiana know. And there's also the economic side of rice production. So most of these farmers are growing rice. Some are harvesting the rice and using it for other things, or selling it for money. And some of these guys are just leaving the rice in the ponds, and that's the base of the food web for the crawfish production. So what is my role in this project? This project for me is kind of just starting up as crawfish season is starting. We had a kind of a cold start, so we're getting ready to jump into this on my end. So what I'm interested in right now is doing temperature monitoring of these ponds to see if I can get kind of a real-time estimation of what's going on temperature-wise, while also monitoring outbreaks of the disease. And so I'm also trying to get some historical data of past outbreaks and see if I can model any patterns of what is happening with this virus. So for me, one of the biggest things is benefiting farmers. And so the impacts of this disease are devastating to the industry. And I really want to connect with stakeholders and use extension professionals through Sea Grant to create resources for stakeholders and producers to understand how we can manage this. So with that, thank you so much for listening to me rant about crawfish, and I will entertain any questions. I have a couple of minutes before I'll pass it on to our next speaker.
I had a quick question, actually.
Robert's question. Yeah, Ariel, please.
So I looked it up online, and it says that the virus doesn't impact like humans, but would it get humans sick if they consumed like crawfish that have been infected with this virus or had this disease?
That is a great question. Thank you for bringing that up, actually. So no, this virus is not transmitted to humans through eating crawfish that might be infected. And that's something that we're really trying to get out there with outreach too, because we want people to enjoy crawfish. And although the production might be slowing down, this virus is not affecting humans at all. And there is no transmission, even if you were to eat an infected crawfish.
Okay, thank you.
Yeah.
I got a question. I got a question directly
Yes.
from Dr. Turner, and it's kind of related to my question, so you could take them together. His question was, how many years has the disease been consequential?
Yeah, so to answer that question, and Robert's question was also that it showed up kind of for the first time in 2007. Was this just in Louisiana? So it didn't just show up in 2007, but there was a study kind of trying to see if this was actually a big problem in 2007. So this has been a problem in shrimp populations for a very long time, and it's more studied in shrimp populations. But we're just starting to see how this could affect crawfish and other copepods and invertebrates. So I think from talking to farmers in my, limited time here, this has been a consequential issue for probably the past 10 years. Maybe this has shown up and they've lost a couple of crawfish, but at this point, whole ponds are being lost, which is a lot of money for these farmers. So in the past 10 years, I think consecutively, this has become a really big problem. So thank you for that question. Awesome. Well, if there are no more questions, I will pass it off to the next person. So next is Jennifer Argote, and I will pass it off to her, and she can give an introduction to herself, but I really thank her for being here today.
Okay, everyone. So I am Jennifer Argote. I'm a PhD student in the Environmental Science Department. I've been around for quite a few years now. I did a couple years on campus and have been doing my work a little more remotely the last few years while I work in the wind and solar development industry. But one of my kind of biggest passions academically is what I'm presenting on today. Title of my project is Reducing Flood Losses with the National Flood Insurance Program and the Community Rating System. And I want to give a quick shout out to my dad, who is actually on this call, sort of watching the struggle that he went through with my whole family during Hurricane Katrina really inspired my interest in flooding and flood insurance and kind of all the work that I'm doing right now. So thanks, Dad. All right. So a little bit more about my project. a lot of the research that I'm doing is based on some work that was started up by Dr. Reams and Dr. Nina Lamb. And I have my committee advisors, Dr. Rivera and Professor Wilkins as well, and got some funding from the LSU Superfund Research Center and the NSF for this work. So right now I'm going to be going over the basics of my research, and then I'll describe the National Flood Insurance Program, the community rating system, and go through a little bit of my preliminary research for my dissertation. All right, so the simple facts that most of us on this call know is that coastal regions are very vulnerable to flooding. Flooding costs money in the form of property damages, economic losses, businesses that are forced to close for months at a time. And the best way to reduce these losses is to try to stop them from happening in the first place. Obviously, we can't just stop hurricanes from happening, but we can prevent, take certain measures to prevent the damages that they can cause. However, not every county or parish is in the same boat in terms of how they can prepare, in terms of money, political views, lots of different factors. So the research that I'm doing is going to analyze the factors that are likely to influence parish and counties' participation in the National Flood Insurance Program's community rating system. So what is the National Flood Insurance Program? So it's a federal program that was started in the 1960s. Right now, over 5 million homes are insured, and the premiums that you pay are based on these things called FIRMs, flood insurance rate maps. And these FIRMs can vary widely based on political factors, based on changing environmental factors. I know in the Denham Springs area, when that flooded a couple of years ago, there's a lot of drama related to the FIRMs because I believe some local politicians had rallied to get the town in a less aggressive flood zone ranking. And then that led to, when the flood actually did happen, a lot of people didn't quite realize what was possible happening in their area. So more on the NFIP. Right now, the program is over $20 billion in debt. When I started my research a couple of years ago, it was $24 billion in debt. So we've improved a little bit. And the majority of the debt from this program is caused by major disasters that happen in large cities. Two of the biggest ones being Hurricane Katrina and Hurricane Sandy, which at the time, Sandy was, I believe, the most expensive federal disaster ever because it affected much wealthier areas in New York and New Jersey that weren't used to this type of damage. And because of that, since they weren't used to that damage and the FIRMs weren't really up to date, a lot of these homes had their premiums heavily subsidized, and that's the case throughout coastal Louisiana as well. The risk that these areas face is a lot higher than kind of the dollar signs point. And in some ways that's important because people can't afford insanely expensive insurance or else they'll move out. And that becomes kind of part of the greater question of, should these people be moving? Should there be better funding to improve their housing so they don't need to move? And that brings up a whole lot of public policy questions. And so the whole point of the National Flood Insurance Program, in theory, is to limit development in flood zones. But in reality, we all love going to condos in Florida, hanging out down at fishing camps and stuff. So from an economic standpoint, that's not necessarily something we want to do. All right, so a state breakdown of the National Flood Insurance policies. So I know these numbers may be a little hard for you to see right now, but it's pretty clear that the majority of policies are in Texas, Louisiana, and Florida. And obviously Texas is physically much larger than Louisiana and Florida, but Louisiana has a very vulnerable coast, as we know, and Florida, of course, is just super exposed to the Gulf and the Atlantic. Okay, so the CRS, this is something I don't think I had heard of till I started my research at LSU. So the Community Rating System is a program within the National Flood Insurance Program that was created in the 1990s to incentivize communities to reduce their flood risk and in turn get lower premiums on their flood insurance. There are a couple different activities that you can do as a participating member of the Community Rating System. And based on those activities, you get a score. that then affects your overall premium reduction. And participation is completely optional, but it can be done at both the county and the town level. So for example, Jefferson Parish participates, but so does the town of Jean Lafitte. That's a town nested within Jefferson Parish. So that's created some slight complications with my data, just looking at different scales of observation. And then my last point here is that costs vary greatly. Some of the activities include building a levee. That can be $1 billion. Other activities can be publishing recent flood, the height of flood waters in recent storms, publishing that. I believe Jefferson Parish does that on water bills, actually. You might not have noticed that, but they do publish it there. That, you know, adding one line of text to a water bill is a variable line item expense. So the cost to participate in different categories can vary a lot. So here are a couple of different categories: public information, flood damage reduction, mapping and regulations. This one, mapping, you know, is one of the simpler ones. You can get points for having books available in a public library that show the different flood zones. The last one, flood preparedness, dam safety, levee safety, that sort of stuff. And so once you complete the activities in this slide, you add all of those up and you're given a set of points that then give you a class from 1 to 10, with 1 being the best. If you have a score of 1, you get a 45% reduction on your insurance, flood insurance. That's on a parish or town level. This goes all the way down to a score of 10, which even if you have a couple small things, and if you don't quite get to the 500-point level, you'll get no points. So I have on this table a couple kind of samples of participating counties. I believe these are all in California. So if you have a class score of 8, you get a 10% discount. But if you improve to a class score of 5, you get a 25% discount. And so that can lead to some pretty big savings in the hundreds of dollars on your flood insurance premiums. All right, so that's kind of the basics of the CRS and the NFIP. So the ability to participate in the CRS is not uniform. Again, we talk about public funding, opinions, your actual storm risk. You know, coastal Louisiana is obviously super vulnerable to storm surge, hurricanes. And then you can look at Florida, where the whole state is super vulnerable, but some of the more inland counties are less vulnerable to things like storm surge. So the CRS encourages risk reduction at a local level. There's no one's forcing you at a federal level. Money is not being withheld if you don't participate, but it's all sort of trying to encourage this community action. And ultimately, if you have improved CRS scores, in theory, that should lead to reduced costs for your flood insurance. And the biggest thing, the whole point of it, increased protection from flooding. So my dissertation, I'm looking at 120 counties along the coast of the Gulf of Mexico. NOAA has a set of coastal communities that are basically all communities that physically touch the coast and go in about 100 miles inland. And so that's just the kind of bar that I'm using for my research. So I'm looking at the conditions under which selected counties are more likely to participate in the CRS and implement these flood mitigating activities. I'm looking at what explains variation across the CRS and why some counties are performing better than others. So some of the factors that I'm looking at are socioeconomic factors, physical exposures, and policy and planning capacity. When I talk about policy and planning capacity, I'm looking at funding for the CRS programs, the number of employees dedicated to coastal planning in a given county or town office. And yeah, so these are just the variables. I'll be running a regression analysis for this to pull the data for the actual dissertation numbers. And on the right here, we have a map of all the participating counties. So it's kind of interesting. You'll see a lot of gray in Louisiana, a lot of counties that are not participating. Then you'll see Florida, almost every county is participating. And so kind of using the if you can keep that image of this map in your head, some interesting numbers that I've pulled already from this data is that only 4 out of 11 counties in Louisiana, along Louisiana's coast, are participating. amongst those counties, there are a few that are participating at the town level, but you might think that Louisiana, being so vulnerable, would have a little bit higher participation. So that's kind of weird. In Texas, there are no counties that participate, or sorry, there's no participation at the county level. So I removed them from my study. There is a lot of participation on the town level, but for the kind of units of analysis that I'm looking at, we just removed them. Sorry, I'm speeding up a little bit. I know my time is running low. Looking at the map of all of the counties, so Florida has very high participation. They also generally have really high scores. If you were to take the 120 counties that I'm looking at and remove Florida, the score would drop down by a whole class. So that's 5% of savings that are wiped out if you remove Florida. And just like a reminder that a higher score is bad. On the right here, I have a map of county participation in over the last 11 years. And you can see there has been a slight uptick from 50% to 57% in the last 10 years. That may be tied to hurricanes and severe flood events that have happened along those areas. So now I'm going to hop into, sorry, I didn't talk about this too much earlier. Back in 2014, led up by Dr. Lamb and Dr. Reams, there was an article published that kind of looked at a lot of these factors, but just for Louisiana. And so my research is expanding this to across the Gulf Coast to those NOAA coastal communities. So here's a little bit of the preliminary results that we got. So higher CRS scores tend to be associated with higher average housing values. The implementation of CRS activities is largely influenced by socioeconomic attributes, and counties with fewer economic resources and lower government capacity face the biggest obstacles. Looking ahead in my dissertation, I'm going to look deeper into the possible influence of planning and policy culture. I'm going to take a look at why Florida is doing so much better. I need to get on the phone with some floodplain managers there. I've been in touch with Jefferson Parish a lot. They have, I mean, kind of as an effect of Hurricane Katrina, they've certainly been hard at work at improving their score the last few years. Yeah, and so future implications of this research is really looking at how we can improve CRS participation to improve outcomes after flood events. And this could be done through increasing funding to programs like Sea Grant and maybe having them help with some of the outreach. But that's just something to kind of look at. And sort of the overall context of this research is just there's so much change going on in the world, and especially in coastal cities. There are a lot of kind of alarmist things going on around sea level rise and increases in hurricanes. But coastal areas tend to be like, people are flocking to cities, they're flocking to coasts. Some stats here are, let's see, first one, 33%, Hillsborough County, Florida, that's where Tampa is. Their population has increased 33% in the last 15 years. Moving over to Texas, Galveston County has had a 27% increase in population. However, the city of Galveston's population has decreased by 12%, which is pretty huge. And that, I believe, is directly tied to Hurricane Ike, which devastated the area. And then below that, you'll see Jefferson Parish, which has still had an overall net loss of 4% of its population since Hurricane Katrina.
Hey, Jennifer, do you think you could
Yep.
restart the share? I think it dropped, and you might want to just share your PowerPoint again.
Ooh, okay.
It happened just a minute ago, so.
Gotcha. Okay. Sorry, that's my last slide.
Just reshare your screen.
Reshare? Okay, one second. Okay, it seems to have kicked me out of the meeting entirely on my laptop. So one second. That was my last slide. I'll just, as I try to pull this back up, my last sort of statistic was just about how sea level is rising. Louisiana is one of the most vulnerable. The Louisiana Delta is statistically one of the most vulnerable places in the entire world, in the Mississippi Delta. And we've just been, these doomsday scenarios that you hear about on the news or that we've been warned about or have had foreshadowing on, some of them are sort of coming into reality, like Hurricane Sandy being an example. I don't think anyone ever thought that lower Manhattan would flood or that a hurricane would cause flooding in a subway. Yet that is what we're seeing. So I'm hoping that my research will help influence preparedness. Oh, sorry, I'm getting reconnected now. Hoping that my research will help communities get more involved in preparing themselves and help reduce losses, help reduce loss of life, and just kind of improve life for everyone here on the coast. So I'll try to pull up my screen one last time, but that pretty much wraps it up.
Okay, any questions? Thank you, Jennifer.
Thanks. Sorry,
Yeah,
for a
thank-
second I wasn't sure if you heard any of that last
No, we
few
did.
minutes. Gotcha.
You did.
Okay.
I was able to follow
was
even
great.
when your slides did drop for a moment.
Gotcha.
So it's
I'll put that last slide up, but yeah, sorry about that.
No worries. Thank you. That was great.
Yeah,
Um,
sorry. I
if
know I was
anyone-
rushing through it a little bit.
No, that's okay. 15 minutes is kind of a crunch to share what you want to share. Awesome. Well, if no one has any questions right now, if you think of any, feel free to put them in the chat, or you can send them to me or Robert. But I will briefly introduce
Oh.
the next speaker, who is Robert Forney, and he is going to tell us about some cool work that he is doing. So I will give him the floor.
I hope it's cool. Thank you. Let's see. I'm Robert. I work with Dr. Paul Miller. This is some of the work that we did together, and I presented this at the AMS meeting, which was, I guess, like a month ago now. Feels like a month ago. And so what I was doing was ranking the urban rainfall effects of Southeast US cities. And here I have just one big slide because this was my poster for the virtual meeting. And over here on the right, I have my final rankings of what the urban rainfall effects were in the Southeast US cities. But before I get into all that, I just want to see if I can give a brief overview of what the urban rainfall effect is before I get into the methods, because if I just jump into like how I got this, it'll be very confusing without a little bit of background. So I'm gonna do something a little bit weird and share a whiteboard and then draw everybody a picture. I hope that's cool with everybody. Okay, can you guys see my whiteboard? Haley, is it showing up?
Yes.
All right, cool. So let's take a horizontal view of, actually, how do I draw on it? I've never done this before. I must have a toolbar here somewhere. Yeah, there we go. Boom, that's my pen. Ooh, it went all black. Oh, okay, there we go. So we're gonna look at a horizontal view, and this is gonna look like an Atari video game or something. All right, cool, that's my city. With a little flag on it, I guess. All right, so that's a city. It goes black every single time I touch the thing, so this is gonna be a lot harder than I thought it would be. So there's this urban rainfall effect, and it's all in the name, right? So there's this modification of the normal rainfall patterns near cities. And the theoretical background for this is that in a sort of normal environment, like far away from the city, the sunlight from the sun is going to heat up the ground, and as the air gets hotter, it's going to rise. And when you have an environment far away that has like, I don't know, like some happy little trees or something, let me see if I can make a couple trees. It goes black every single time. This is very frustrating. All right, so this is gonna be a little tree. So far away from the city, you got trees and stuff, and they absorb a lot of heat themselves because they're full of water, and water has a high heat capacity. But near the city, it's mostly like concrete and glass and metal and things like that, which have a much lower heat capacity. So the area near the city will heat up a lot more easily. And so it'll rise more, and we get a circulation as a result. It's a lot like the sea breeze. So if you're familiar with the sea breeze pattern, it looks a lot like it. Like we have air that rises over the city, right? It's going up. This is very disconcerting how it goes black every single time. All right, so it goes up over the city, and then it will create a circulation where it returns at the ground level. And as a result, because of the circulation, it comes towards the city at the level of the ground approximately. This is very, very funky. It goes up and it comes back. And wherever you see these up arrows, you're going to get uplift of the moist air. And this is going to cause some thunderstorms that happen specifically in the locality of the cities. You're not going to see this kind of thunderstorm activity away from the city. And when you add some kind of large scale advection like this, so there's some kind of mean flow, it means that you're going to see enhanced thunderstorm activity downwind of the cities. So it'll be less on the left side here, be more on the right side, because that's the direction of the advection. So this is what I'm studying. And this is actually a very complex thing, but this is kind of the basis of it. So coming back to my poster, we would like to know what the amount of urban rainfall effect is for each city. It seems pretty intuitive, I hope, from the diagram that a bigger city or a more well-developed city will have a more significant urban rainfall effect, but it's hard to parse out what fraction of the normal thunderstorm activity is from the city and what is from the background, like what normal amount of activity there might be. Because thunderstorm activity, it varies with latitude, it varies with other environmental factors, proximity to the coast. So I was hoping to try to get at least a first order estimate of what the activity is of thunderstorms from cities. It's also worth mentioning that the thunderstorms can come in a variety of sizes and dump different amounts of rain. But one of the assumptions that I made in this analysis is that the thunderstorms that are caused by urban effects are all dropping relatively the same amount of rain, and they're all around the same size. That means they all have the same reflectivity when you shine radar on them. So I had access to this cool big 15-year data set that Dr. Miller and his co-authors developed in 2017. Up here in the top left in the introduction box, I have a roster, which is just showing how many times a weakly forced thunderstorm, WFT, the kind that's from urban heat island effect or urban rainfall effect, how many times they crossed over those locations. So a hotter color like red means there's more of it. So you can see at the end of Florida, there's a lot going on there. In some more remote regions far away from cities and far away from mountains, you don't see as much. They're more blue, meaning there's fewer thunderstorm crossings. So that's the data set I'm working with. That's what that roster is kind of representing. And the way that we identified the thunderstorms was we took the radar reflectivity, which it gets everything. It'll get airplanes and buildings and whatever, ground clutter, everything, hurricanes, thunderstorms. It gets everything in there. And then we kind of cleaned the data to create a data set that's just the weakly forced thunderstorms. And that's what I was working on. And here in the data methods section, there's a flow chart that shows how we created these composite images of the reflectivity, where we get like a footprint for each storm that crosses over our domain. And then we assigned some attributes to them so that we could manipulate the data, say like how many thunderstorms happened in a certain area, what regions did they cover, how long were they in each place. So using that information, like the footprints of each thunderstorm and how many there were, I was able to just count up how many are within a certain range of each of the cities. We did like 32 cities, all in the southeastern US, where we see a lot of these weakly forced thunderstorms. And for each city, I put down a buffer. So did this in ArcGIS, which is admittedly not my forte, but a buffer is like, it's a region on the map over which you are making some statistics. So we did a standard radius of 100 kilometers around each city as kind of our control. That's like the average. So when within 100 kilometers of the city, it'll include how many thunderstorms went across the area that's not city, as well as the area that is city. So that's kind of like the average case, the background. And we also did a smaller buffer, which was 20 or 35 or 50 kilometers, which is just over the city and the immediate area around it. So we were kind of expecting to see more of these thunderstorms in the smaller buffers that are just over the city relative to the overall larger area. So we took ratios of the smaller areas over cities to the larger area, which includes the city as well as the surrounding environment. And then we just ranked the cities according to what ratios we saw. So a higher ratio means that we see more weekly force thunderstorms per unit area in the smaller region than in the bigger region. So that's how we ultimately got this table here on the right. I'm just gonna need to move all my Zoom stuff, sorry. Yeah, so over here on the right, we have this table. It's called Table 3. It's in the results and discussion. And so you can see in the top, there's a lot of cities from Florida that were all highly ranked. That's kind of not surprising. I mean, just looking at this roster, figure 1, you can see that a lot of thunderstorms crossed in the lower part. Okay, so Alexi's saying we have, okay, never mind. Okay, so someone's saying the poster wasn't showing well. Anyway, sorry about that. So anyway, so in this table, the higher values means there's a higher urban rainfall effect ratio that we calculated. And you can see all the Florida cities are very highly ranked. And there's just 3 that were very lowly ranked. And these 3 actually made a lot of sense because they were technically large in terms of their spatial extent, but they have low populations. They're not well developed. So it's not necessarily the case that just because they are large cities that they will have a significant effect. One thing that was kind of funky and interesting is that Atlanta didn't rank that highly. Atlanta has been very well studied and is sort of the poster child for Southeast US urban rainfall effect cities. You know, it has a very big effect, and it ranked only 11th on this list. So I think that this first order attempt at ranking the urban rainfall effects is pretty decent, but probably it could be improved by using a more intelligent buffer design, maybe something that follows the direction of the mean wind, because we're expecting to see some enhancement downwind, and I like slapped a big circle on it. You know, it was not keeping in mind that there's sort of a shape in which we would see the enhanced urban rainfall. And another thing that is significant is that a lot of these cities are really near the ocean. In the roster in figure 1, you can see that over the ocean, there's not really that many weekly force thunderstorms, just because they don't have the kinds of properties that kick up the warm, moist air. It's very flat and smooth. So this method could get better, but it's at least a step in maybe the right direction. So with that, I'll take any questions that you guys have. That's pretty much my whole spiel. Another factor that's important that I failed to mention is that orographic features like mountains can cause weekly force thunderstorm enhancement as well. So another thing that could be done to enhance this could be to do a little more cleaning of the data set, maybe to mask out areas where we can't do a very rigorous analysis. Questions, comments? Dr. D'Elia mentioned that in the St. Petersburg, Florida area, there are definitely a lot of storms like this. And he said that it seems that it's common from the stall in the TPA area. I'm sort of acronymically challenged. Dr. D'Elia, if you could unmute and tell me where's TPA, maybe we could discuss a little more. It does seem like maybe the thunderstorm stalling could be an urban rainfall effect there.
TPA is Tampa Airport.
Oh, okay, so it's Tampa Airport. I had some interesting thoughts about well, I hope they're interesting to other people, but I found it interesting to think about what it means for there to be so many WFTs in that area too, because there are just so many effects that can lead to enhancement in that area. It's hard to parse out which one is urban rainfall effect. Like you have convergent sea breeze in that area as well. You know, you have a sea breeze from the west side of Florida, sea breeze from the east side of Florida, and it all just kind of comes together in the middle. That could also lead to it. I thought that maybe the stalling could have to do with something like that, but it could also be urban. So probably I'll need a smarter analysis technique to separate the sea breeze from the urban effect, but that's a good question. Thanks for clarifying. Okay, I don't think I have any more questions, so back to you, Haley.
Awesome. Well, thank you, Robert. That was great. And last but definitely not least is Sophie Jurgensen, and I will give it to her now to wrap us up.
All right. Hi, everybody. Thank you, Haley, for the introduction. So I'm going to be discussing the first chapter of my dissertation. I'm a third-year PhD student working with Dr. Jennifer Brum in the Go Viral lab in Docs. So I'm going to be discussing what we've learned about some viral communities in a marine oxygen minimum zone. So a little different style of talk, and also more about one than just one virus. So I want to start off by stating that we're living in a microbial world. So microbes dominate all biomes on Earth numerically and are driving the biogeochemical cycles that fuel life, including carbon, nitrogen, and sulfur cycling. And we are focusing in here on the bacteria in the ocean. And one of the main controlling factors on bacteria is the viruses that infect them. So most viruses in the world infect bacteria, and oftentimes in the environment, they outnumber their hosts by about an order of magnitude. So we like to say that there's about 200 million viruses per mouthful of seawater, to give you an idea of the numerical dominance of viruses. And they're also quite active. So there's an estimated 10 to the 23rd viral infections per second in the ocean. And these viral infections have pretty vast ecosystem impacts. The most obvious of which, of course, is that viruses serve to convert happy little bacteria into dissolved and particulate organic matter through the process of lysis, so killing the bacteria. And this contributes to recycling of organic matter within the ocean. So I study viruses in oxygen minimum zones. These are areas of the ocean with little to no measurable oxygen at depth. If you were at Walkeel's Defense this morning, you might have heard a little bit about those. So this is a global map showing oxygen concentration at 200 meters depth. So all of the areas that are in red or pink have high measurable oxygen, and the areas that are in cooler colors and these dark blue have little to no measurable oxygen, or it's below the detection limit. And sometimes you might hear these referred to as dead zones, but actually these areas are full of microbial life. And a lot of the microbes in these areas are also doing all kinds of unique metabolisms that contribute dramatically to the cycling of particularly nitrogen in our atmosphere. The viruses in these areas are pretty understudied at this point. So far, there have been papers published looking at the viral communities here in the Eastern Tropical South Pacific off the coast of Peru, and also in Cariaco Basin. And what I'm going to be discussing with you is our study looking at the viral communities of the Eastern Tropical North Pacific, which is here off the western coast of Mexico. So for the duration of the talk, I'm going to be discussing data we have from two different stations. One is more offshore and it'll be designated by these triangles, and one is more onshore and will be designated by the circles. So the first step that we took to investigate these viral communities was to look at their abundance in the environment. So to do this, we use a technique called epifluorescence microscopy, where both bacteria and viruses are filtered together and stained with a special chemical that binds to DNA. And then we can visualize them together and enumerate them. So in this case, the bacteria are these larger green dots, and viruses are the really tiny pinpricks of light in the background. So doing this, we're able to look at the relative abundances of viruses in their hosts with depth. So I'm going to be showing you several depth profiles throughout this talk. So I just want to orient you to how these are all going to be set up. So here, in terms of these graphs, I have the viral and bacterial abundances on the x-axis here. This y-axis is depth. Like I said earlier, the shape of the symbols corresponds to the station that the samples are from, and their color corresponds to the depth category in the water column, which we've determined based on oxygen concentration. So just for these depth profiles, I have oxygen concentration in this light gray for each station in the background. I know it looks a little cluttered, but that's just for this to orient you. The future depth profiles won't have that on there. But so here the green symbols are representing the mixed layer samples. So that's where oxygen is high. All of our red symbols are representing the oxycline, where there's a sharp decrease in oxygen concentration over a short vertical space. So that's represented by this really sharp, almost horizontal gray line. The blue symbols represent samples from the OMZ core, where there is oxygen is below the detection limit in this case, so it's here hovering at 0. And our yellow symbols down at the bottom are representative of samples that are below the OMZ, so they have relatively low oxygen, but it is above the detection limit. So just looking at the data here from our abundances of these two different microbes, we can see that both viruses and bacteria have a peak in abundance in the mixed layer. At both stations, there's obviously a much larger peak for viral abundances in the nearshore mixed layer. And there is a secondary peak in bacterial abundance in the upper OMZ of the nearshore station. But overall, we're seeing this peak in the surface ocean and then a decrease in abundance with depth. Next, we wanted to see if these viruses that we can see with our epifluorescence microscopy are actually actively infectious. So we use transmission electron microscopy. So using this technique, we can visualize bacterial cells. So here's a bacterial cell, this little squiggly thing. And sometimes you can see viruses actively infecting bacterial cells. These are the darker dots within a cell to get an idea about the frequency of viral infection. So when we look at these data, we see a pretty similar trend for both stations where we have a peak primarily in the mixed layer at both stations, and also a secondary peak in the frequency of infection within the OMZ core. So these data are providing evidence that viruses are both abundant, so present, and also actively infecting hosts within the OMZ layer of this area. So next I'm going to be discussing some of our sequencing results. So I'm going to very briefly introduce you to the concept of metagenomics, which is how we're looking at the DNA of these viral communities. So the overall concept is to collect and filter water samples to isolate viruses. Then we extract the viral DNA, chop it up, and sequence it. We take those little chunks of DNA that we've sequenced and assemble them together into these larger assembled sequences. And at this point, we can start to look for presence and absence of those different sequences in any sample. Also look at their relative abundance. We can classify them as viral or not, cluster them into populations, which is similar to what you can think of as a bacterial species. So you can think of them as a population is kind of like a species. And we can also investigate their genetic composition. So what genes do these different populations have? And we can guess as to why. So if we start just looking at this viral community structure based on those populations that I was just discussing, the first thing I looked at was the presence and absence of those populations in all of our different samples. So what just popped up on the screen is called an Euler diagram. You can think of it as like a Venn diagram here. So the circular area is representative of how many samples are shared or unique to each of these different areas. So in this graph, both of our stations are combined. And you can see that most of the populations are either unique to the mixed layer, so unique to this green circle, which is oxygenated, or are unique to the oxycline, or shared within the oxycline, OMZ, below OMZ, all of those different low oxygen samples, some combination of those. So we're starting to see a pretty clear delineation between mixed layer samples that have oxygen and low oxygen samples. I next looked at the relative abundance of these populations using a form of ordination called correspondence analysis that also takes into account environmental data. So in this case, all of the symbols are representative of a sample. So this green circle represents the mixed layer nearshore community. And the distance between any of the symbols in both the X and Y directions indicates the differences in viral community structure, so the magnitude of that difference. So for our case, the mixed layer samples are separated based on station, and all of the low oxygen samples are combined together. And one of the most driving factors for this analysis was determined to be oxygen. So to understand if there were differences between those low oxygen samples, since in this visualization, they're all just on top of each other, I next removed the mixed layer samples from the analysis and looked at just the low oxygen ones. And when we do this, you can see that the samples are clustering based on their color, which is their depth category, where they are in the water column, rather than their station. So this is providing more evidence that both depth and oxygen are the major driving forces of structuring the viral community in this area. Our next step was to start looking more at the genetic composition of these different viral populations. And one of the main things that we started to look for are called auxiliary metabolic genes. So during the course of viral infection of a host, the virus hijacks the host metabolism primarily so that it can have the host produce more viral DNA and also all of the virus parts, so the capsid and tail. But also during infection, viruses can sometimes have auxiliary metabolic genes that are sometimes called host genes that can adjust the host metabolic trajectory in a different way. So these can be have been identified to be involved in different regulatory processes or nutrient cycling. So I'll just briefly go through one of the most classic examples of auxiliary metabolic genes involves photosynthesis. So if you imagine a happy little prochlorococcus cell in the surface ocean where there's high light and it's capable of doing photosynthesis, one of the genes required for this process is called PSBA. If a virus with PSBA successfully infects the host cell by injecting its DNA, that DNA can actually be expressed and speed up the process of photosynthesis. And I don't want you to think that this is the virus trying to be nice and make the cell happy and have more energy and materials for itself to grow and replicate. This is a viral strategy so that the virus can steal those extra energy and materials so it can replicate itself faster, kill the host, and then move on to the next cell. As you can probably imagine, this is not a strategy that is limited exclusively to photosynthesis. So viruses can encode all kinds of genes related to lots of different metabolic processes, including genes involved in nitrogen and sulfur cycling and other regulatory processes. There's a lot of different auxiliary metabolic genes that have been identified. So we investigated the composition of auxiliary metabolic genes within the Eastern Tropical North Pacific samples that we have. So if we're just looking at the total auxiliary metabolic genes that we were able to identify, on the x-axis here is the normalized abundance of those genes. So that's based on the sequencing depth. You can see that both stations follow a pretty similar trend where there is relatively low amount of these AMGs in the mixed layer that then increase dramatically in abundance in the oxycline and then begin to decrease in depth from there. So this provides some evidence that maybe viruses are utilizing different strategies in the oxycline that would require more of these different AMGs. We first looked for those classic AMGs that we've heard about previously, like photosynthesis genes, and we did detect several viral encoded photosynthesis genes within our mixed layer samples, which makes sense because that's where photosynthetic organisms live. So it makes sense that the viruses infecting them would be there. But when we started to look for other types of functional categories like nitrogen and sulfur cycling, which we know are important in marine oxygen minimum zones and have been documented to be occurring in particularly the ETNP, we see basically 0 genes related to nitrogen and sulfur metabolism within our viruses. And those that we do see are mostly regulatory. So this gives us some evidence that viruses are probably using a net different strategy in this area of the ocean. So we started to dive more deeply into what different functional categories were present in these metagenomes, particularly what was abundant in these low oxygen samples. And what we saw was a lot of genes involved in the biosynthesis and formation of iron sulfur clusters. So these are iron sulfur clusters are really important prosthetic groups in a lot of different metabolic reactions and are very easily inhibited by differences in oxygen concentration within a host. We also saw an increase in sporulation genes. So sporulation is a bacterial kind of defense strategy against unfavorable conditions to allow the bacteria to continue to persist. And toxin antitoxin genes, which are more well characterized in plasmids, but are a form of addiction system for the host. So we noticed that all of these different categories follow the same general pattern in terms of their peak abundances in the oxycline with our total AMGs. And important to note here that all of these can be related to oxygen stress for dealing with oxygen stress for the host bacteria. So what we think is happening in this area of the ocean is that viral strategies, or the main viral strategy utilized by the community, differs based on oxygen concentration. So in the surface ocean, where there is both high oxygen and light, we think we're seeing this more classic story where viruses are using the main net strategy of boosting the host metabolism in the upper ocean so that they can exploit more energy and materials generated by the host to then kill them and move on to the next host. But in the OMZ, where there's low oxygen and some of the hosts are under stress from this lack of oxygen, we think that a main viral strategy is to try to keep their hosts alive for long enough to viruses to replicate at all in this oxycline and OMZ core. So it's a net switch within the general community. So I hope that you've seen from this brief talk that viruses in the Eastern Tropical North Pacific are abundant and active within the OMZ layers of the water column, that viral community structure is strongly driven by oxygen. Key differences between the viral communities are largely due to their auxiliary metabolic gene composition, and that viral strategies may switch from boosting metabolism to protecting hosts from low oxygen stress within the oxycline and OMZ core. And with that, I'd like to just acknowledge my funding sources and take any questions. I can leave you on this.
Thank you, Sophie, for that. That was that was great. Thank you. If anyone has any questions for the four of us, feel free to reach out to the seminar's email, and we can put you in contact with us directly. We would love to talk more if other questions arise. With that, I really appreciate everyone joining in today on this Friday to let us present and share some of our work with you. Thanks for bearing with us as we dealt with technology issues in the current virtual world that we live in right now. But with that, I hope everyone has a great weekend. Thanks so much.